I would like to start with thanking the organizers for having me and being able to present our project here, which is indeed about MS-based proteomics data in the context of protein groups and networks. And it's a joint project with Marie-Lucas Poulet from the University of Toulouse, Scooter Morris from University of California, San Francisco, and Lasio Jensen, in whose lab I am. And you might recognize, especially in this audience, you might recognize these names from something that is a little bit different, which is namely String App, which is one of the more popular Cytoscape apps. And I would like to just mention a few words about it. Namely, our most common use case is that people have a proteomics data, or it could be other omics data, with a list of uniprot identifiers, some experimental measurements. And then what they do is to take these identifiers, go to string cytoscape, create a network, and also, because this is one of the advantages of having the data in cytoscape, actually map the experimental data onto the network to see the relationship between these nodes, and also in the context of up and down regulation. So proteomics data is something that we have to deal with quite often. And for those of you in the audience who have already analyzed it, you might be aware that protein identification is quite a challenge, especially in bottom-up mass spectrometry. What we basically do is that we have a sample, we digest the proteins into peptides, put them through the mass spec, and then we want to actually find out which were these proteins that were in the sample to start with. In order to do that, there's various software tools that would map the peptides back to the protein accessions, the protein sequences. And what happens sometimes is that we have cases. Okay, no mouse. Never mind. So we have cases where we have basically unique peptides that can be mapped to the protein, so we can identify this was the protein in the sample. However, in many cases, we don't actually get a peptide that can uniquely identify a protein, as it's the case in the second example, where we have two different peptides that map to two different proteins. So there's no way, basically, to find out what was in the initial sample. And what these tools do in these cases is that they report protein groups instead of single protein accessions. And I have an example here, which is basically one protein group that contains one uniprot identifier and another one with two uniprot identifiers for the two proteins. What is common then at some follow-up analysis is that people would like to see what are the biological processes that are related to these proteins, maybe build some networks, as I showed in the previous slide. So basically, they do some of these functional enrichment analysis on networks. And the problem is that these tools usually operate on a gene protein level annotation, so they cannot take protein groups as input. So what happens is that basically people would often just take the first identifier of each protein group and do the analysis with that, which is not exactly a very proper handling of these protein groups. So because of that, we set out to investigate in different studies, basically, what kind of protein groups do we have and how this strategy of choosing only one protein identifier from each protein group can impact the downstream analysis. We had several questions. One of them was, okay, what are the proteins in the protein groups? Are they similar or different to each other? How this choosing only one identifier impacts functional enrichment analysis, network analysis, and the last but not least, can we do something? Can we suggest some best practices? Can we provide maybe some solutions? In order to do that, we selected 14 different data sets. And here I give you an overview. I know it's a bit small, but basically we have several different organisms, these publications from different research labs, different years in the last 5 to 8 years. And also we try to cover also different technologies. So we also have single cell proteomics, we have phosphoproteomics, and so on. And also different accession reporting strategies. So if we look at this data, it would be interesting as a basically first insight to see how many of the what is the proportion of protein groups that have these multiple accessions compared to the ones that have only one accession. And the multiple ones here are in black. And you see that for some data sets, it's even more than half of the protein groups contain multiple accessions. What I should note is that this is the original data that we took from the supplementary tables of the different publications. What we did as a next step is to map these identifiers in each of the protein groups to identifiers with which we can work to do functional enrichment analysis and build networks. So in our case, we mapped them to string identifiers. And due to partly the way that protein groups are reported and what data is used to search with, we might have several different isoforms for protein, or we can have some ambiguity in identifiers. So what happens is that these multi-member protein groups are reduced to a certain extent, but you can still see that there's black bars in most of the data sets. And we are now in the range of tens to hundreds of protein groups with multiple members compared to before being in hundreds to thousands. So the first question was, how similar are the proteins in the protein group? And in order to assess this, we looked at two different measures, namely the sequence similarity and a way of looking at functional similarity based on gene ontology annotations. And I'm not going to show you the plots, but if you're interested, just find me. I'll just give you the conclusions that we drew from this analysis. And the first one is that, as expected, protein similarity was very strong for proteins that are in the same protein group in terms of sequence. This is not unexpected because in order to be in the same protein group, they actually share part of the sequence. For the functional similarity, we can say that still proteins in the same protein groups are more similar to each other if you basically look at the distribution of all the pairs in the dataset. However, we see many more cases where there's a difference in the annotation. It could be that one protein is annotated and another one is not annotated at all, or still there's some difference. So basically, yes, proteins can have a different set of functional annotations. So now knowing this, we ask ourselves, okay, what happens when we do functional enrichment analysis where we basically use these gene ontology annotations to see if any of them are overrepresented in our set of differentially expressed proteins or other set of interest. And in order to evaluate this, we did the following. We basically, for each protein group, selected randomly one of the accessions, one of the genes to represent it, performed gene set enrichment analysis using these same gene ontology annotations, and kept the significant enriched Go terms. And we didn't do that only once, we did it 10 times, just to have an approximation of what happens if you basically do it randomly a few times. What we get out of this is a plot that looks like this, where basically on the x-axis, we have the number of enriched Go terms. And on the y-axis, we have the proportion of the rows in which this Go term was identified as significant. And what that means is that you see this big bar at 1. This means that the majority of the Go terms were in all 10 of the iterations. So it seems we are quite robust. Of course, you can see that there is cases where a Go term shows up only in one iteration or only in a few of those. But looking at this plot and several others that we did, so we looked specifically at 2 of the studies in terms of functional enrichment, we can conclude that picking only one of these proteins from the protein group doesn't impact so strongly the functional enrichment as one might have expected or not, depending a bit on what you expected. Okay, so the next step was, okay, what happens when we do network analysis? And here I have a bit of a more visual example. We have 2 different protein groups in this network, highlighted in orange. And you can clearly see that depending on which protein you pick from the protein group, you have a very different network connectivity. In each group, there's one protein that is not connected at all, and then there's 2 proteins that have a slightly different connectivity. What is nice is that we can actually quantify what is kind of the chance, the probability of having a protein group as a whole being connected to the rest of the network. In the lower example, if you look at the edge between CPV1 and TAC3, you can see, okay, there's only one edge between this protein group and this protein, while there's two proteins in the protein group. So basically, we have a chance of 50% to have the group being connected to the network. On the other hand, if you look at the two protein groups, one with the two members and one with three members, there's only one edge connecting these two protein groups, while there could potentially be 6 different edges connecting all the different members. So we have 1 out of 6 chance to basically have this group being connected, these two groupings connected. So what we decided to do is basically for each data set, we created a string network, a high confidence network, and calculated this probability for all the protein groups. What we get out of that is a plot that looks like this. So it's a bit similar to the enrichment one with some differences though, and I'm showing here all the data sets. And what we have here on the x-axis is basically this probability of the protein groups being connected. And on the y-axis, you see basically the proportion of such groups. And the striking thing is there's 2 main peaks in these plots, and you see it's very consistent across data sets. The first one is at probability of 1, and these are basically all the protein groups for which all the protein group members have the same network connectivity. So it doesn't matter which one would pick. The second peak, which is at 0.5, mostly results from protein groups that have 2 members, and we have quite a lot of those. And in these cases, basically we have different connectivity. Most likely one of the proteins is not connected to the network while the other is. But you can also see there's a bit of bars that are below 1. So if we kind of summarize this information across all data sets, we have up to half of the connections of protein groups that have multiple members that would be affected by what we choose. So now the question is, can we do something about that? And knowing us, you might have a guess already. So we developed a small app for Cytoscape called ProteoVisualizer, which basically uses the string app and wraps around it in order to create string networks taking into account the protein groups. How this works is that basically the user can input their list of protein groups that basically comes from the table outputted by MaxQuant or whatever search engine they use. They can also decide what kind of string network they want and also supply some options for the protein groups. What we do then is that we have some kind of consensus network taking into account the protein groups, representing them as one node in this network, and basically also aggregating the node and edge information for the protein group members into this representative node of the protein group. And what you maybe see is that some of the lines are dashed. And this is a way for us to highlight when the confidence basically of the interaction decreases, because not all of the group members are actually connected to the node. So one might be interested in actually seeing, okay, how is this one protein group, which are the members of this protein group that are connected to CSF1R? And in order to do that, you can actually just double-click, expand the group, and see what are the connections of the proteins to this specific node that is not in the protein group. What is also quite common, and I showed on the first slide in proteomics, is that we would like to also see these networks in context of the data that we have. So you can still easily do that, as I'm showing here with just an example of, for the proteins that are in this network, of their up and down regulation in a comparison that was done in the publication. And you can go even a bit more fancy. And in case there's multi-omics data or there are different comparisons done in the study, you can use an app called Omics Visualizer that we also developed and actually map different comparisons to the same nodes. In this case, we see that they behave quite similarly, although in one of the comparisons, the regulation is a bit stronger. So to conclude, we basically see that protein groups can be handled much better when performing downstream analysis. Just picking one accession at random wouldn't be the recommended best practices. Is not exactly the solution. For the more proteomics people of the audience, I would like to say that taking the leading or master accession is much, much better than taking all accessions. We elaborate on that more in the manuscript. And we show that this is less of an issue for functional enrichment, but definitely more for networks, which is why we provide this Cytoscape app that you can find on the Cytoscape App Store and use to create networks that are aware of protein groups. I would like to acknowledge Marie, Scooter, and Lars, without whom that work wouldn't have happened, and all our affiliations and funding. And thank you for listening.
Thank you, Nadia. We have time for a few questions. Go ahead, Anne-Claude.
This was great. Thank you very much. So protein groups are not really stable across experiments, right? And for people that don't do proteomics, they tend to be worse if the coverage is lower, right? And it tends to be a bit better if the coverage. So how do you propose to the framework to do that? Like if you have different experiments that you want to compare, do you propose to create kind of a master protein group first before using your tool, or?
So when creating this network, we went with the assumption that basically you have already done some kind of analysis of the data and compared the different experiments. We see that a little bit in the phosphoproteomics data, where we basically can have different information mapped on the protein group than on the individual proteins. And what is nice in these networks is you can have this expanded version of the protein groups, and you can actually map the data onto it and see the differences.
Okay.
So I would expect something similar to happen if you have different experiments. You could have kind of a summarized protein groups, but then basically still keep the information about the subgroups. But it might be something that we need to test with and maybe accommodate a little bit. But I think there's
I'm just thinking because between experiments, at least in our hands, the stability is not there, right? So the group, what is in the group is not constant, depending of the coverage, providing that the sequences are not in fact identical, right?
Yes.
So thank you.
I'll take one more question here. Yeah.
So I guess maybe on the same topic, would an hypergraph representation help seeing the differences in the different experiments? So one could have an hypergraph that encompasses a given a given protein group, and then another one that would have a subset of that, and one that could be distinguished, and then one could therefore investigate the graph to see what are the real master protein group, as Dr. Jaga was referring to.
I suspect that's possible in Cytoscape.
You can, yeah. You can.
Yes.
You can do hypergraphs in Cytoscape.
Yeah. I haven't thought about it. I think it's definitely interesting to look into and try out. Even already with these groups, which we use in Cytoscape to implement that, you could actually have different visualizations. So one option is what I showed, where you have basically the bounded box around the proteins. But you could also have just all the proteins connected to the protein group node.
Right.
So maybe that would be a way to also represent it as a hyper. Yeah.
Thank you.